famous Shanghai mountain, and it's very beautiful. And on the top of the mountain, there is a Finch Lake. We also call it as the Heaven Lake. So this lake will not frozen all the year around, even if it's on the top of the mountain. So I like the plants, I like the wildlife, but I never thought that the life gave me a gift to make me a plant biologist. Actually, I start my plant biology study, start from my master study. I finished my master degree in the Chinese Academy of Agricultural Science in Beijing, China. And during my master study, I studied the plant pasture interaction. I used the rice as my research material and to study the mechanism of how rice resistant to the bacterial infection. And during this project, I found amazing gene can increase the resistance of the bacterial infection for the rice. After that, I joined the Domestic Life Science Laboratory in Singapore, and I did the biofuel project. And this is a commercial project. We used a kind of plants called the jatropha to use the seeds to produce biofuels. So this project is collaborate with Toyota company, and is very amazing project. And after that, I come here, join the Dr. Enamel Hart's lab at the University of Texas, Austin, and now I'm mainly work on studying how light regulates the plant development. So today I will share the story about how light regulates the life of the plants. Um, we all know that light can provide food and energy for plants through a process called photosynthesis. So the photosynthesis is the plants use the sunlight, carbon dioxide, and the water to produce oxygen and the sugars. And those sugars can provide fuel and energy for plants to develop. Do plants use light only for food and fuel? Yes? Or no? Actually, that's not all. So, the light can regulate the plant development throughout the plant life cycle. So, from the seed germination and seedling deaculation and seed avoidance, chloroplast movement, and stomata opening, and the circadian clock, and even the phototropism, and the flowering time. So you can see here, the light cannot only provide the food and energy to the plants, but light can also regulate the whole life cycle of the plants. And there are some more strange words here that you might quite understand about that. And later on, I will give more details about those processes. Okay? First, let's see the seedling deaculation. What does deaculation mean? Actually, it talks about a process. We all know that seedling plant life starts from the seeds, right? We bury them into the soil, so the seeds grow start from dark. And after that, plants can grow out from the soil, and their life transitions to the light. So the deaculation talks about a process that the plant life transitions from the dark to the light. Okay? And did you know what dark brown seedlings look like? No. Actually, I prepared some dark brown seedlings, and if you are interested, I have 4 plates, and who would like to check those plates and tell us what did you see from these plates? And those are dark brown seedlings. You want to try? Yeah, I have 4 plates. If you are interested, you can take one of them to check.
Any volunteer from this side?
What did you see from the plate?
Very small black dots.
Small black dots?
Little string also hang down.
Yes. And what did you see?
There without color.
Cover. Yeah. How about without color? Yes, we have here. Sorry. Yeah. But it's good. Yeah. Why don't you tell us? Yeah. Any yeah. Who can tell me? What did you see?
You guys want to share?
What did you see? Yeah. Do you want to tell me what?
Yeah, I saw some white strings with young black.
I'm being
I will
honest.
show you guys what you will see from the face.
Yeah, I'm gonna say that.
Okay. So, in the place, you might see the seedling like this, right? They have a very long stem. Actually, it is a elongated head portal. In the plant biology, we call this stem like structure called as the head portal. Why in the dark they have such a long, elongated head portal?
Looking for light.
Yes, good. Yeah, that's correct. Because under dark, plants cannot do the photosynthesis. They need food and energy. So they need to grow as soon as possible to the surface of soil to approach the sunlight to make food and fuel. And also, you will see a closed apical hook. And this structure is called the apical hook. And also, you can see a closed yellow cotyledon. Why the apical hook is closed? And why the cotyledon is also closed? Why? No light. Yes. Yes, no light. And yeah, protection. Yes, that's correct. We know that when the seedling grow upward, they will have some mechanic damage for the leaf. And for this area, it caused the cotyledon. Between these two closed cotyledon, there is an area called the shoot apical hook. So this area is a self-renewal area. And after the seedlings grow out of the soil, all the upground tissues will grow up from here. So the closed apical hook and the closed cotyledon will protect this area from damage when they grow upward. Okay? So this process in the plant biology, it caused the scotomorphogenesis. So like this, when you plant sunflower seeds under soil, you can see it has a longer head portal and a closed apical hook and a closed cotyledon. Okay, so this process is described the seedling development under the dark. Okay, so today you learned a new word. It's called scotomorphogenesis. Okay, and also after the seedling approach to the light, what the seedlings will look like. I also prepared some plates. Okay. Who would like to check those seedlings? It's super nice. Yeah. What did you say? Is it the same with the dark green seedlings?
It's like when it goes to the like, the process of like,
Is it the same with the dark green seedlings? Who would like to tell us what did you see? What did you guys see?
I think he
It's
saw
okay,
something.
yeah.
I think I did.
Just to
Yeah, I
try.
think you did. Yeah, needed. What did you see? I can't really notice.
Okay. Did you see the seedlings look like this? Yes, right? So you can see it has a shorter head quarter. Okay, why? Now the head quarter becomes shorter, but not longer anymore. Yeah, it's exposed to the light. And now the plants will focusing on develop the leaf and the roots for getting the more sunlight and get more nutrition from the ground. And so they will not waste the energy anymore to elongate the head quarters. Okay, so now you can see the open apple hook and the open the green cotyledon. So this process is called photomorphogenesis. So you can see the seedlings grow out of the soil and they open the apple hook and open the cotyledon. So the photomorphogenesis describes the process that plant grows under the light. Okay? So today you learned 3 new words. Dehydration, scotomorphogenesis, and photomorphogenesis. You don't have to remember those words, but one day if you talk with a plant scientist, you will scare them. Okay. Next, we will talk about another interesting part. It's called seed avoidance. So seed avoidance actually is a smart strategy for the plants to maximum their absorption of the sunlight. And what is the seed avoidance? Actually, is a responses when the plants sense the seed from the other plants. So they will use some smart method to overcome these seed conditions. Let's see how they overcome those conditions. Okay, so before the response, all the plants grows together, close with each other, but there is no enough sunlight. There are some seed conditions from neighbors. So some plants, they are smart. They grow faster and harder. They overtop neighbors, and then they can perceive more sunlight, make more food and energy. Okay, and this is one method. Another method. So you can see here, they can shift the position away from the seed neighbors. Very smart, right? So those seed avoidance actually you can also apply to the agriculture practice, right? Now when you want to plant some tomatoes at home, you need to leave some space for each plant to help them perceive more sunlight. Okay? Yeah, so this is talk about the seed avoidance. And we all know that plants cannot move. But do you know plant chloroplast can move? No. Okay. So what is the chloroplast? Actually, the chloroplast is located in the plant cell, and they are the location where the photosynthesis takes place. And those green structures is the chloroplast, and those chlorophyll are the pigments inside of the chloroplast which can perceive the sunlight. And those green chloroplasts actually can move. Can you see those green dots? They are moving under the light. Okay? See this green part?
Why do they move?
Yes, where they are born and how. Actually, in the plant cell, there are some cytoskeletons. They can help those chloroplasts to move, but those movements actually regulated by light. Let's see how it is regulated by light. Actually, those movement also called the chloroplast relocation. Okay, so you can see under the low light, that there is not enough light, the chloroplast will try to accumulate on the top of the leaf. Okay, so this will help them to pursue more sunlight to produce food and energy. But another condition, which is higher light, it will destroy the photosystem and also destroy the chloroplast. So the plant want to avoid this happen. So you can see the chloroplast will move to the side of the cell. And under darkness, those chloroplasts will accumulate on the bottom of the cell. Why? They will save the energy, right? They want to save the energy, do not waste the energy, because under dark, no light cannot provide the food energy, and they will accumulate at the bottom of the cell. This will help them to move to the good position as soon as they perceive the sunlight. Okay, so this is the chloroplast relocation. It is regulated by light. And another part I really like about to talk the phototropism. The photo, you can see this word. Photo, that means the light, and the tropism, actually it talks about a trance. Okay, so the phototropism, it means the plant grows towards the light. Did you see this before? Always, right? If you put your flower nearby the window, the flower will grow towards the window. But actually, it is not grows towards the window, it's towards the light, right? And here I have a question. If you want to to make your plant grow vertically again, what will you do? Perfect. Yes, you just turn this side to the window, right? And after some time, it will grow vertically again. Okay. So the phototropism actually is the binding of the plants towards the light. Now let's talk about the mechanism of how phototropism occurs. Before we talk about that, we need to know the plant hormones, like oxygen. Oxygen can help cells elongate or inhibit the elongate of the cell. It depends on the situation and the concentration of the oxygen. And in the phototropism, if you put your seedlings under the direct light, the oxygen will secreted from the tip of the plants and distributed from the top to the bottom. So all the cells will elongate at the seedlings. But if you put the seedlings under the indirect light, you can see all those options will distribute on the seed side. So the seed side options will help the cell elongated faster than another side. So the plants can bind towards the light. Okay? So this is how plant hormone regulates the phototropism. And this process is also regulated by the light. Okay. So for the flowering time, how light regulates the flowering time? Actually, in plant biology, there is another word. It's called photoperiodism. So it talks about short-day plants and long-day plants. How these plants flower. So let's say the short-day plants. So for those kind of plants, if you put them into the day length over 12 hours, they will not flower. But if you put those short-day plants into the day length less than 12 hours, it will flower. And if you put those short day plants under even shorter daylight, but a longer darkness, during the darkness, you give some of the light. The plants still cannot flower. That's so interesting, right? And for the long day plants, they need longer daytime. So when you put the long day plants into a daytime longer than 12 hours, it can flower. But if you put it into a day length less than 12 hours, it will not open. But if you put long day plants into a day length less than 12 hours, but the darkness over 12 hours, but during the darkness, you give some lights, the flower still can open. Okay, so this is very interesting. Okay, so let's talk about the importance of a photoperiodism. So for the one important thing is this is a seasonal adaptation. So the plants will flower at the most advantageous time for the pollinations and seed productions. And they also can be applied to the agricultural practice. Like we can use this to optimize the planting and harvesting time, and we can also use this mechanism to improve crop yields and qualities. Okay, and let's see the long day plants. We talk about the long day plants. They need the daytime longer than 5 hours. Okay, so what kind of plants belong to the long day plants? The beets and potatoes, lettuce and radish, they are all belong to the long day plants. So when you plant those plants, you can choose the late spring to early summer to plant those plants. Okay, and for the short day plants, their flower when the night length exceeds 12 hours. Okay, so what kind of plants belong to the short day plants? Soybean and rice, tobacco and cotton. Okay, so when you plant those plants, you can choose early spring or late summer to early fall. And another different one is called day neutral plants. So for these day neutral plants, they flower regardless of day length. So that means they can produce flowers and fruit continuously, like cucumber, corn, sunflower, and tomato. If you want to plant them anytime, all the year. Okay, so you can get those fruits. Okay, so it's so interesting, right? So how light regulate the plant development? Can plants see light and respond to it just like what we do? Can plants see light? How? See it. Yes, sense it. It can feel it. Actually, in the plants, there are some specific proteins called photoreceptors. Okay, so those proteins can perceive the sunlight and transform these signals to the plant and tell them, oh, I perceive this sunlight. You need to give some response. Okay, and so those photoreceptors can perceive the UVB light, UVA, blue light, red, and infrared light. Okay, so for the UVR8, they are the proteins which can sense the UVB light. It can accumulation to the UVB light, and it can also regulate the stomata opening, and also including the phototropics. Photo 1, photo 2, they are all proteins. They can perceive the UVA or blue light. So the phototropins, they can regulate the tropical growth, just like regulate the gravitropism and phototropism. And they can also regulate the stomata opening, chloroplast movement. And for the love domain containing F-box proteins, those kind of proteins, they can also sense the UVA or blue light and regulate the flowering time and the circadian clock. For the crack crops, those ones can regulate the deethylation, flowering time, and the circadian clock. And for the phytochrome, actually they can sense the red and the far red light. They are very important to regulate the plant development. So they can regulate the seed germination, deethylation, gravitropism, flowering time, and the circadian clock. Okay, so I will use the seedling deethylation as an example to talk about how light regulates the cotyledon opening. And here we use the phytochrome as an example to talk about the mechanisms. Okay, so we talk about the phytochromes. They are the proteins can perceive the red light and the far red light. So there are two cells. This one, green one, is the cell under the dark. And this yellow cell, it means the cell is under the light conditions. So you can see under dark, these phytochromes are green color. Actually, they are not active. They do not work. They are lazy. Okay. But inside of the nucleus, there are a group of proteins called transcription factors. Those transcription factors can start the transcription process to inhibit photomorphogenesis. But at the same time, they can promote the scotomorphogenesis development of plants. So that's why under the dark, you can see the seedlings have a longer hypothetical closed apical book and a closed cotyledon. But as soon as the cell perceives the sunlight, those phytochromes, they are inactive. But when perceive the sunlight, they are weak. They can work now. And then they can move into the nucleus to further phosphorate those transcription factors. And those phosphorylation will be recognized by the E3 ligase. And the E3 ligase can add the ubiquitination to those transcription factors to make it recognized by proteasome. Those proteasome can degrade those transcription factors. They become to small pieces, cannot work anymore. So they will promote the photomorphogenesis. So now you can see open the cotyledon. So this is a mechanism for how phytochrome regulates the cotyledon opening. I hope this is not too much. Okay. So and maybe you also wonder, how do we study the mechanism by which light regulates the plant development in the lab? So what's the lab work we do? So actually with the faster development of the technology, previously we used the chemical measures or physical measures to identify regulators. But now we use the computational based method to identify those important regulators. Okay, so we can identify lots of regulators involved in response to blue light, response to oxygen, and photosynthesis, and response to far red light and red light. So after we found those regulators, we will do some biology studies to see their functions. How to study their functions? First, we will check the phenotype of those genes. So here, I take A gene as an example. So under dark, you can see here is the white type. No any changes for this kind of plants. But for A-1, A-2, and A-3, actually we take out this A gene. So in those plants, no A genes. Okay? So those seedlings shows the same phenotype compared with the white type. But when we give more A gene into those seedlings, we express more A genes and more genes, more A genes. So these seedlings, you can see, they have an open top needle and an opened apical hoop. Okay, and under red light, you can see the wild type, they have bigger cork needle, right? But 8-1, 8-2, 8-3, which do not have A gene, they will show a smaller cork needle. Okay, so actually those are mutations of the A gene. Okay? And if they overexpress A gene, you can see all those overexpression have an even bigger cork needle opening compared with this white type. Actually, A gene is insulinase. It can positively regulate the light-induced cork needle opening in oral pulses. Okay. So how A gene works in this pathway? We talked about the ubiquitination of those transcription factors, right? So the ubiquitination will be recognized by protease, and those proteases can degrade those transcription factors. Actually, the A gene is the enzyme which can ubiquitinate these transcription factors. Okay, is that too much? Yeah, because those are related to a lot of biological methods and the biochemistry method, so I will not introduce too much about that. But this is what we do in the lab. Okay, so if you are interested in, you can contact with us, and even if you can visit our labs. Okay, so that's all what I talked today, and thanks for my supervisor, Dr. Yanan Bai, and thanks for all my lab members and all the people who helped me a lot. And also thanks for all of you to be here, and I hope you guys learn something and have some fun. Thank you.
Questions? Anybody? Questions?
Thank you. That was a great session, I think. I was wondering about the effect of excessive light, if the genes that are related with this kind of stress, if some other stressors like drugs or maybe many other factors because of environment can affect or express the same genes that one plant can produce or release in order to save themselves.
I want to repeat your question.
I'm sorry.
I
It's
just
not
want to make
I'm not
it.
quite sure what to do.
I will repeat it. So, do you want to ask me if I overexpress this gene will affect other pathways or?
Sorry. I mean something like if the genes that are going to be expressed when you're exposing the plant with sunlight or excessive light are also expressed because of other stressors that might be interacting with the same plant.
Yeah.
Maybe the lack of water activate the same genes for
Yeah,
the
we
plant.
will. Yeah, actually, in the lab, we will control those conditions. We have the specific light source chambers only provide the red light for this pathway to study these gene pathways. And we will control the temperature, humidity, and the medium content. All those conditions will be the same.
Oh, okay. My question was going to be in order to understand what happens in real life without controlling.
Oh,
some-
yeah, that's a very good question. So under the in the real life, we will see a lot of crossing words if we mutate this gene or if we overexpress this gene. Actually, during my study, this gene also shows the response to the heat. heat stress. So if we want to study this gene in the light signaling pathway, we will separate it to two parts. And for the heat stress, we will study it separately with the heat conditions. And if we want to study its function and the light, we will separate it with other pathways. But just like what you say, if we want to see the if it can be applied to the agricultural practice, we will do some field work to do the test.
Thank you.
I'm sure you said this at some point, but how does exactly the movement of the plant hormones cause the physical bending in the phototropism?
Oh.
Like, I kind of missed that part. Is it?
Yeah, actually, if you put the plants under the direct sunlight, the oxygen will produce on the top. It will produce on the top.
Oh, okay.
So it will distribute downward.
Thank you. Sorry. How exactly do you remove or like overexpress the genes in the plants that what you have? Is there like a tool, a tool you can use to do that? How
Yes.
is that done?
It's all related to the molecular biology method. We use a kind of agribacteria to infect the plants. So this infection will transfer a vector into the plants to overexpress those genes.
And so by overexpress, you mean just putting more copies of the gene in
Yes.
the genome?
Correct.
Thank you.
Mhm.
Do you have a resource you could share that maybe shows like a chart of the different plants and their flowering time?
Oh, yeah. If you like to get those resources, I can post on the Facebook, maybe under the science. I will share with them and they can post it on the Facebook. Mhm.
How does heat affect? It seems really related to the light.
Yes.
And I only saw one variable that mentioned coldness, but it seems as if heat might be.
So actually the heat stress also related to the related to those photoreceptors. And we talk about the phytochromes. Actually it also involved regulate the plant response to the heat stress. So it has a molecular mechanism for how to those phytochromes to regulate the heat stress. If you are interested in, I can also post those resources to the Facebook. Any other questions?
The video that you showed of the chloroplast moving, how fast does that happen in plants?
How fast? Yeah, it depends on the light intensity.
Okay.
Yeah. So the light intensity can regulate those movements. If it is under good conditions, it's not moving too fast. It's just focusing on I get the energy. I don't want to move too fast to consume a lot of energy.
So like, could it be, if it's a lot of light, could it be minutes or hours? Like what is the time scale like?
Wow, this is like a college exam. Yeah, actually I didn't know those correct numbers, but if you are interested, I can check it at the later. Yeah. And other questions?
How common is it for one gene to perform the same function across different plant species?
Sorry, I cannot hear you.
How common is it for one gene to perform the same function across multiple plant species? common is it for one gene to perform the same function across multiple plants?
It is actually it varies from species to species. Like this gene represent tree guys, it only present in the rice and orango. For other species, no crystal. And for the animals, they do not have it.
Any more questions? Any questions from the back? All right. Thanks everybody for coming. Our next event is not going to be until September, so be on the lookout for our fall 2025 schedule around July or June somewhere. All right. Thanks everybody for coming. Have a good, wonderful evening.